Hi class, Dr. Jim here. In today's lecture, what we're going to be looking at is a special kind of cell division, which we call meiosis. Meiosis is really important for two types of cells that we have in our bodies. Whether you're male and you make sperm, or whether you're female and make eggs, this is where meiosis takes place. With meiosis, we do a special kind of cell division where we actually reduce the number of chromosomes in half before we divide our cells. And this is really important because when you have the sperm and egg come back together, you want to make sure you have the right number of chromosomes. Okay, and so that our babies don't end up with the wrong number, we want to make sure we get the right one. And so in order for that to happen, we do this process called meiosis. All right, so let's take a look and go on the journey of what meiosis is all about. All right, so our objectives today is first is why do we have to reduce the numbers? I've already alluded to one of the reasons why, because we don't want our babies to have the wrong chromosome number. So we have to reduce the numbers in order to ensure that our children or our offspring get the right number of chromosomes, which would be 46. Okay, so we'll look at that. Why do we have to reduce the number? The second thing is we're going to talk about the special type of cell division known as meiosis and go through the steps. Now you're going to notice that a lot of the steps seem very similar to what we did last week with mitosis. And I want you to kind of keep mitosis in the back of that and in the back of your head because we're going to use some comparisons and contrasts of mitosis and meiosis, especially to explain some of the differences that you see. All right? And we're going to look at these differences between mitosis and meiosis. And again, this is something you're going to need to know for the test. So even though you've been tested on mitosis and you probably have that somewhere crammed in your head, somewhere in your cranium, we really want you to know the differences between mitosis and meiosis. And we think that's fair game to ask you those things on this next test. Now, even though you've already taken the test on mitosis, that doesn't mean to forget everything you know about those things. And so we'll go through step by step and look at some of the differences. And so we want you to be able to know those differences and why these things occur. The last thing we're going to look at is why do we get genetic variation. And so the reason why we do meiosis in the first place is to do something called sexual reproduction. And sexual reproduction is a very good thing because it allows our offspring to have genetic variation. If we only did asexual reproduction, and if you remember that from mitosis where some animals do this, basically you make a clone of the adult. And so the child looks exactly like the adult, which is fine when you want to establish large numbers of population. However, if something happens in the environment that eliminates one of the offspring, all of them are going to be eliminated because they're all the same. But if you have genetic variation, it allows you to have better survival. You're able to adapt and survive. And so let's give the example of, let's say you change the temperature of the environment by dropping it 20 degrees. Those organisms that have adaptations that allow them to survive at colder temperatures will survive better than the ones that don't. And so that's where we see the strongest will survive or the best genes win out. And so genetic variation is very important, and that's why we do that. That's why we do sexual reproduction. Okay? So that's a good point to know, and we're going to see why we get it or how we get it through this process. All right, so why do we have to have the number? Well, I talked about this already a couple of times. But again, you want to have your sperm to have half the number or the haploid number of your chromosomes along with the eggs so that when you fertilize, the baby has the correct number. If we left them at 46, what would you have? You would have a sperm that has 46 and an egg that has 46. They come together and the baby would have 92. That's not a good thing. You don't want to have more genes than what you're supposed to have. And there's in the next lecture, you're going to see what happens when the chromosomes don't divide evenly, and it causes some genetic disruptions and problems that we have to deal with. Okay, so in order for things to work properly, we have to reduce the number of chromosomes in the sperm and eggs before they come together and fertilize to make a new offspring, and so they have the correct number. Okay, so that's really important. Why we reduce? Because you want to have the correct number in the baby. All right, so we call this meiosis, and meiosis, which I like to refer to as the reduction division, because the first set, which is always described as meiosis 1, is the reduction part, where you reduce the number from 46 to 23. And then the second part, which is referred to as meiosis 2, is the cell division part. And so we're going to look at these things and look at some of the differences and things that go on during each of the phases. But I want you to think about reduction division. You have to reduce the number of chromosomes before you can divide your cells. Okay? It's used only for sexual reproduction, so the only place you see this is in your gamete cells, whether you're a male, you make sperm, or the female that makes eggs. And this is the only place that you see it. Everywhere else is normal mitosis. This is the only place that you see meiosis. And again, what happens is you have the number prior to fertilization, so that basically you have your parents, which are diploid, okay, both have 46. They reduce the number to 23, the haploid number, if you remember that from last time, the haploid number. And what you get is when they get fertilized, you end up with a baby that has 46. That's the important part. Same number of chromosomes as parents. In order for that to happen, you have to reduce the number or have the number of chromosomes. Okay? So if we look at it, we've talked a number of things about chromosomes and things like this, but now what we're worried about is the pairs, okay? So we're talking about the pair from mom versus the pair from dad. So you get one chromosome from mom and one chromosome from dad, and that equals a pair, okay? And we call this the homologous pair because they look very similar, and they might be very similar, but they're not exactly the same, okay? Because depending on what genes you have on what, they're going to be very similar in what genes they have, but the types of genes or the varieties, think of like an ice cream cone. It's all the same ice cream cone, but you have different flavors on those things, and those different flavors give you the genetic variation. All right? And so we call those two a homolog because you have the one that looks like mom's gene or has from mom, and the one that looks or comes from dad, and they look exactly alike except one's from mom and one's from dad, and that's called a homolog. And you can see down here, this is what they would look like before you go through replication, that S phase or synthesis phase. You do replication and it comes out with the X. And you can see here, we have 22 somatic pairs, okay? That represents all the different genes and all the different chromosomes we have. And then we have our 23rd pair, which determines our sex, either XX or XY. And we get one of these from mom and one of these from dad. Okay, and so we'll talk more about genetics and where we get those things in the upcoming lectures, but for now I want you to be just aware of the different homologs and that you get one from mom, one from dad, and that total is 46 or 23 pairs. Okay, so that's where we're going. Alright, so if we look at meiosis as an overview, we want to see what happens in the cell. Now if you look at the cell itself, what you can see, this is before they go through replication and make that duplicated chromosome. But I want you to count the number. You have 2 chromosomes, okay, 2 total chromosomes, but a total of 4 chromosomes, I should say. 2 from mom, the red ones, and 2 from dad. Okay, so you have a total of 4. After replication, you get those X's, and what happens during the process of meiosis is they line up. And when they line up, they don't line up all in one long line, but they line up as their pairs. And I'll talk more about this as we get into the specific phases. We then divide and we reduce the numbers. So you can see after that first set, remember meiosis 1 that I talked about earlier, you go from having 4 chromosomes to now having 2 in each cell. So that's the reduction part. You then divide the cells again to actually end up with 2 more cells. And so a total, you end up with 4 cells, but each one of those cells has 2 chromosomes. And if you remember back to mitosis, what did you end up with? You ended up with 4 chromosomes with 2 cells. In this situation, you end up with 4 cells with 2 chromosomes. Okay? And that's really one of the biggest differences between mitosis and meiosis. But we're going to look at some of the phases and look at these specific differences as we go along. Okay? So, like I said before, we distinguish meiosis into 2 separate, distinct parts. Okay? The first part is called meiosis 1. It's designated by the Roman numeral 1. This is the reduction part. This is where we go from 4 chromosomes to 2, or in humans, we go from 46 to 23. Okay? Very important. So we have the same names of the phases. They're just distinguished by the Roman numeral. Either your prophase, metaphase, anaphase, or telophase 1, or your prophase, metaphase, anaphase, telophase 2. Okay? So that's what we're looking at. In prophase 1, which is the start of meiosis, we see that the nuclear envelope fragments and you lose it. It disintegrates just like it would in prophase of mitosis. The chromatin ball condenses into the chromosomes. But what's different about this one is that your homologous pairs find each other, and that these homologous pairs kind of bind together and they sort together. And we call these things, they line up side by side, and we call this a synapsis when they line up side by side. We refer to these things as bivalents. So in your book, they call them bivalents. When I went to school, we always referred to them as tetrads. So you can think of it however you want, either a bivalent or a tetrad. I will probably refer to them as tetrads because that's just how I learned it. But in your book, they call them bivalents. It means the same thing. So bivalent, tetrad, interchange for either one. All that means is you have the chromosomes, the homologous pairs, the one from mom, the one from dad, next to each other, and they're mingling together. Okay, and that's what happens during prophase 1. Now in metaphase 1, what happens is now the cell blows the whistle and says line up. But instead of all the chromosomes lining up in one long line, they actually line up as their pairs. So in our cells, we would actually have 23 pairs of chromosomes lining up as opposed to having all 46 line up in one long line. And that's a big difference because now we're talking about the pairs. We're going to pull apart the pairs rather than the individual chromosomes right now. Okay, so the bivalents line up independently of one another. And what you can see is some of moms might line up on one side and some of dads might line on the other. And again, this is all independent. There's no rhyme or reason. It just happens. Okay, so that's the good thing. It adds to the variation. Now the next step that happens is called anaphase 1. And in anaphase 1, instead of splitting the individual chromosome apart, so remember the duplicated chromosome and the chromatids getting pulled apart, now what you're doing is pulling the homologous pairs. So you're either pulling dad's chromosomes to one side or mom's to the other or vice versa. And again, the mix. And so what you do when you split that cell is you're basically going through and splitting the number of chromosomes. You're going from 4 chromosomes in our example here. So remember before they are in mitosis you had 4. Now that you start to split the cell, you're reducing that number to 2. So that's the important thing. You reduce the number. So in humans, you go from the 46 down to the 23. Okay, reduction part. All right, and then in telophase, which you end up with, again, you get the nuclear membrane reforming. You get the chromosomes that show up in each one. And so what you end up with is now 2 cells with how many chromosomes? 1, 2. 1, 2. Okay, and so that's important. You've reduced the number. You've gone from 4 chromosomes from the start now to 2. Just like in humans, you would start with 46. Now you reduce each cell to 23. Okay, the important step. And that's meiosis 1, dealing with prophase, metaphase, anaphase, and telophase 1. Okay, the next stage is now where we go into the second part, the cell division. And the first thing that happens is what is called interkinesis. And interkinesis is kind of like the timeout part of this division process or meiosis. It's kind of the halfway point, halftime. And in men, the cycle goes actually very quickly because they reproduce sperm very fast. In women, this can be a very long, drawn-out process. Actually, in women, interkinesis is basically kind of that part of cell cycle where you go into suspended animation. So meiosis 1 actually happens in women during development as they're developing in mom's belly. And then by the time that they're ready to have children of their own, they go through meiosis 2 right at the onset of fertilization. And so really that interkinesis is a very long, drawn-out process for females. In males, it's very quick. Okay? So just think of it as a process for the cell to have a timeout, halftime. All right? And that's interkinesis. Now we get into the second phase of meiosis, and meiosis 2 involves the cell division part. And you're going to look at this and say, hey, wait a minute, I think I've seen this before, and it looks very similar to what we see in mitosis. Okay, so think of the meiosis 2 as the same thing as mitosis. All right, and so we start with prophase 2. Again, the nuclear membrane disassociates, and you get the spindle fibers moving, or the little centrioles moving to the poles. Okay. They move to the poles, and then these guys line up. Now remember, instead of having 46 in each cell, you only have 23. So you have 23 chromosomes that line up in one long line on the metaphase plate, okay, and they get ready to divide. Now in anaphase, they have that division, so you can see here, now the duplicated chromosomes get pulled apart, and now the individual chromosomes get pulled to either end. And so that's what happens here. You're not reducing the number because each cell is going to have equal numbers. You can see this cell is going to have 2, this cell is going to have 2. Likewise, this cell will have 2, and this cell will have 2. All you're doing is getting ready for cell division, so just like mitosis. All right? And then finally, in telophase 2, in cytokinesis, you now divide the cells. And you can see now you end up with 2 cells up here that have 2 chromosomes and 2 cells down here. So the ultimate goal of meiosis is to end up with 4 cells with half the number of chromosomes. So in humans, we end up with 4 cells that actually have 23 chromosomes. Okay? Important. You want 23 so that when the sperm and egg come together, you make 46. Okay? If it doesn't divide correctly, that's when you have your genetic issues. All right? And so that's the process. So we can look at the differences between mitosis and meiosis, and here's basically it all written out. And this is found in your book, and you can look at this, and it's also in your lab book, looking at the differences between mitosis and meiosis and what happens at these different stages. Now, think of meiosis 1 as a reduction. You don't reduce the number in mitosis. You reduce it in meiosis. And so you can look at all these different things, the bivalences versus all of them lining up. So you have the 23 pairs lining up versus the 46. Here you have the bivalents that separate versus the individuals separating. And then finally getting 2 daughter cells. You go through the second phase of meiosis, which looks very similar to mitosis, but again, now you're talking about numbers. And again, in these cells you have the 23 versus in these cells you have the 46. What I like to show is the pictures, and people like pictures to see what actually is happening. So up on top you have meiosis, on the bottom you have mitosis, and so you can see the differences. So in meiosis, prophase 1, you have that intermingling of the homologs, okay? The intermingling and crossing over takes place, and I'll talk a little bit about crossing over in just a minute, okay? In metaphase 1 of meiosis, you actually have the pairs lining up. As opposed to all of them lining up in one big line, you actually have the pairs. And so remember, 23 pairs in humans versus all 46 lining up in one line. Then you separate the pairs out. You reduce the number. You go from 46 to 23. In this example, it's showing 4 going to 2. Okay? In mitosis, you maintain the number. You start with 4, you end with 4. And that's the big thing. All right? You look at meiosis 2, you divide the cells again, and what the ultimate outcome is 4 cells with 2 chromosomes, whereas in mitosis you have 2 cells that end up with 4 chromosomes. And again, they look identical to what the parent cell looks like. As opposed to meiosis, where you have genetic variation, you have very different looking daughter cells compared to the parent cell. All right, and so those are the big differences. So I can write these out for you, and so you have all the major differences. So let's look at the 2. First, meiosis requires 2 divisions. You have meiosis 1 and meiosis 2, where mitosis, you only have the 1 cell division. Okay? You have chromosomes that synapse, that come together, the pairs come together, they mingle, and they have crossing over, whereas in mitosis, you do not have that going on. Meiosis, you have the number of chromosomes. You go from 46 to 23. In mitosis, you save that number. You stay at 46, and you go from 46 to 46. Okay? At the end, you produce 4 daughter cells. At the end of mitosis, you produce only 2. Okay? You produce cells that are genetically different in meiosis as opposed to mitosis. They're exactly the same. And then finally, this is only used for sexual reproduction, whereas in mitosis, this is used for asexual reproduction in those organisms that do it. But typically, what we use them for is growth and repair. All right? So the next thing I'm going to bring up is what happens in males and females. It's slightly different, so you want to pay attention to see what happens. In males, we go through the process called spermatogenesis, or making of sperm. Okay, and we do basically the process of meiosis and what we talk about. We start with one giant spermatocyte. Okay, we go through meiosis 1, where we end up with 2 cells, and finally after meiosis 2, we end up with 4 mature sperm. The big thing is we go from 46 to 23 in each one of the sperms. Okay, now in females, we call this oogenesis, or the genesis of the oocyte. Okay, and what happens is you start with the primary oocyte, but instead of having 2 cell divisions, you actually only have 1. You have a cell division, but the one egg cell takes up all the cytoplasm and leaves the other just a little bit left. And we call this little bit left the polar body. Now you remember, meiosis 1 stops, you get to that interkinesis, and in females this happens during development in mommy's tummy. Once you've passed that stage, you go into suspended animation until you're ready for your own fertilization of your own offspring. So this could be a number of years, at least 15, 20 years, hopefully, that you don't have any children. But if whenever that happens, that's when you go through the process of meiosis 2. Okay? Go through meiosis 2, you get fertilization, and you get the second division of meiosis 2, where now you end up. And again, you'll get one giant egg cell and end up with 3 polar bodies. So really the end result is that you get one big egg cell, okay, that is 23 chromosomes, and then you get 3 polar bodies that remain with the egg cell that basically are wasted and break off and eventually get reabsorbed, and you lose those. Whereas in sperm, you make 4 mature sperm, all with 23. Once these two fuse together, the sperm and egg, you now have a zygote that ends up with 46 chromosomes, and that's the ultimate goal with meiosis. Reduce the number, then get the sperm and egg to come together. Okay? The other thing is, men start making sperm at the process of puberty and continue on to the rest of their lives, where women, they have a limited supply of eggs. And the reason for this is because of this cell division. You divide 4 cells, but only 1 egg cell is good as opposed to the 3 polar bodies. So you're limited in how many you actually have. And so that's a big difference. Okay? So the last thing we're going to talk about is genetic variation. And genetic variation is the key to adaptation and surviving. And so 2 ways that we get genetic variation is, one, by crossing over. This is when the homologs intermix, and what you end up with is that you get some of dad's chromosome on some of mom's chromosome. As you can see here, here's mom's chromosome, and part of dad ends up on mom's chromosome. Likewise, with that intermingling, some of dad's chromosome or mom's chromosome ends up on dad's chromosome. Gives you some genetic variation. Very cool. More variation, more adaptation ability. Okay. The other thing is the independent assortment. And we're going to see this again when we talk about Mendel and his plants, but this is really important. You can have your chromosomes line up however they do, and this is all independent of what the cell feels like the day it undergoes meiosis. You could have all of dad's on one side and all of mom's on the other side. You could have a mix of more of mom's on one side and more of dad's on the other, and so on and so forth, and you get all these different combinations. These different combinations give you a mixing of the genes, and this is that independent assortment. Mixing is good. Genetic variation is good. Allows you to adapt. And so that's the other source of genetic variation. Not only do you have crossing over, but you have the mixing of genes. And let's look at it just by the raw numbers. Okay? So when you have fertilization, you think about all the different possible types of genes you could have. Well, in humans, we have 23 pairs, so that allows us to go from 2 to the 23, and you figure that you get that same variation from the other person donating the sperm or the egg. And that gives you 70 trillion different possible combinations of the genes itself. Okay? And that's not even thinking about crossing over. So just that alone gives you that many possibilities for genetic variation. If you look at crossing over makes that even bigger changes, and so you can see how big this number is, and I don't even know how big that is. It's just a huge number, and so think of it that way, just a really huge number with crossing over. And that's important because we want to have genetic differences, and allowing us to do sexual reproduction and going through this and having the genetic differences allows us to have different genes for us to adapt to different environments. Again, reducing the number, mom or dad and mom makes baby that has the right number of chromosomes, and that's really important. Okay? So to summarize, meiosis is the process of a special cell division in which you first reduce the number of chromosomes and then divide the cells. We call this reduction division. This is important because you want to make sure your offspring have the right number of chromosomes. If you don't reduce, you don't get the right number of chromosomes, that leads to genetic abnormalities and issues, and we're going to look at that in another lecture. Okay? The differences between meiosis and mitosis, one of the big differences is with meiosis, you end up with 4 cells, and those 4 cells are genetically different. And the other big difference is that you have the number. Instead of having 46 chromosomes, you end up with 23. And then finally, with genetic variation is very important. You get that with crossing over, and you get that with independent assortment. And these are important to allow for variation, to have adaptations so that you can survive different environments. It makes you all look different. So if you're lucky enough to have siblings, you can see that you all look slightly different, have different traits and things like that. That's due to that genetic variation. You don't all look alike. All right? And so we've come to the end of this lecture. I know it's kind of complicated and looking at these different things. Make sure you draw pictures, go through these things step by step so you know what happens, because again, we'll test you on knowing what happens at each step. Make sure you practice, and understand what happens with these things. And if you have any questions, feel free to ask, all right? And we've come to the end. Thank you for watching, and I'll see you next time, all right? Good luck.